Welcome back to Chapter 10, Lipids. In this lecture, which includes part 3 of this chapter, we will discuss about the function of lipids as signals, cofactors, and pigments. Steroid hormones are oxidized derivatives of sterols. They have the sterol nucleus, but they lack the alkyl chain found in cholesterol. They're more polar than cholesterol, and steroid hormones are synthesized from cholesterol in gonads and adrenal glands. Steroid hormones move through the bloodstream from their site of production to target tissues where they enter cells and bind to highly specific receptor proteins in the nucleus and trigger changes in gene expression and thus metabolism. And this is one of the major functions of steroid hormones. Because hormones have very high affinity for their receptors, only very low concentrations of hormones are sufficient to produce responses in target tissues. The major groups of steroid hormones are the male and female sex hormones and the hormones produced by the adrenal cortex, cortisol and aldosterone, these two hormones. Prednisone and prednisolone are steroid drugs with strong anti-inflammatory activities mediated in part by the inhibition of arachidonic, which we'll talk about in subsequent slides. The biosynthesis of cholesterol and other steroid hormones involves multi-step synthesis involving various different enzymes. But the major component or the building block of all sterols and steroids is isoprene. It is these isoprene units that are combined together to form various sterols and steroid hormones. Biologically active lipids are present in much smaller amounts than storage or structural lipids. And in addition, they play vital roles as signaling molecules between nearby cells. Some of the biologically active soluble lipids involves vitamins, vitamin A, D, E, and K. Vitamin A1 or all-trans retinol and its oxidized metabolites, all-trans retinoic acid and retinol, act in the process of development, cell growth, differentiation, and vision. Vitamin A1, as shown here, or beta-carotene in the diet, can be converted enzymatically to all-trans retinoic acid, a retinoid hormone that acts through a family of nuclear receptor proteins to regulate gene expression to embryonic development. That's the process, that's the function of all-trans retinoic acid. Whereas the function of vitamin A1 or all-trans retinol is in the vision pathway. In the visual pathway, this molecule, all-trans retinol, is converted to 11-cis-retinal in an oxidation process. This 11-cis-retinal then combines with a protein named opsin to form what is called as rhodopsin. And this rhodopsin is a visual pigment widespread in nature. In the dark, the retinol of rhodopsin is in the 11-cis form, as you can see. When a rhodopsin molecule is excited by visible light, so when light falls on it, it undergoes a series of photochemical reactions that converts the all-cis to all-trans retinal. And what happens is the transformation results in a neuronal signal. And This signal is then perceived by the brain as a visual transduction. Vitamin D3, also known as cholecalciferol, is normally formed in the skin from 7-dehydrocholesterol in a photochemical reaction driven by the UV component of sunlight. So when you get exposed to UV light from sun, this molecule gets converted to this. The reaction that is happening is the breaking of this bond that is shaded in red. Vitamin D3 is not itself biologically active, and it is converted by enzymes in the liver and kidney to another molecule called as calcitriol. Calcitriol controls rate of calcium absorption from gut and reabsorption from kidneys. Low vitamin D3 leads to diseases such as rickets, which can cause bone deformities and osteoporosis. Vitamin E, K, and other lipid quinones are antioxidants. Vitamin E is the collective name for a group of closely related lipids called as tocopherols, all of which contain substituted aromatic rings and a long isoprenoid side chain. And by the way, this is the basic building block of these vitamins. Now, the aromatic ring, as shown here, reacts with and destroys the most reactive forms of oxygen radicals and other free radicals, protecting unsaturated fatty acids from oxidation and preventing oxidative damage to membrane lipids. And thus, vitamin E functions as antioxidant. Talking about vitamin K is another antioxidant, but it has another function. It is a blood clotting cofactor. The aromatic ring of vitamin K undergoes a cycle of oxidation and reduction during the formation of an enzyme called as active prothrombin, which is a blood plasma protein essential in blood clotting. And vitamin K functions as a cofactor for this protein. So vitamin K is necessary for blood clotting. That's the bottom line here. Warfarin is a blood anticoagulant. It's an FDA-approved drug that inhibits the formation of this protein, prothrombin, and prevents blood clotting. It is also an invaluable anticoagulant drug for treating humans at the risk for excessive blood clotting. For example, in the case of SARS-CoV-2, it was found that one of the major reasons for death is because of blood clots. And warfarin has been experimented as a drug to treat these clots. So warfarin can be used to treat patients, those who have coronary thrombosis. Finally, ubiquinones and plastoquinones are isoprenoids that function as lipophilic electron carriers in oxidation reduction reactions. We'll talk about ubiquinone in detail in oxidative phosphorylation chapter. Lipases are enzymes that catalyze the hydrolysis of lipids. The human pancreatic lipase is a main enzyme that breaks down dietary fats. Dietary fats involve triacylglycerols, which gets broken down to monoacylglycerols and fatty acids. Phospholipases are enzymes that hydrolyzes phospholipids, especially ones in adipose tissues. There are various types of phospholipases, as seen from this image here. Phospholipase A1 cleaves the SN1 acyl chain. This means that phospholipase A1 cleaves the ester bond between the hydroxy at C1 and this fatty acyl chain. Phospholipase A2 cleaves the SN2 acyl chain, which means it cleaves the ester bond between the hydroxy and the fatty acyl chain that is attached to carbon 2. And this specific esterase or lipase is found in cytoplasm and also found in venoms. Additional phospholipases involve phospholipase B that cleaves both SN1 and SN2 acyl chains, or they're called as lysophospholipase. Phospholipase C cleaves before the phosphate. If this is the phosphate group, phospholipase C cleaves before the phosphate, releasing a diacylglycerol. So if it cleaves this bond, you will get a diacylglycerol and a phosphate attached to a head group. And in this case, phosphatidyl inositol 4,5-bisphosphate, you end up with this inositol phosphate and this diacylglycerol chain. PLC plays a central role in signal transduction, releasing the second messenger inositol triphosphate, or IP3. PLD, on the other hand, cleaves after the phosphate. So this bond is before the phosphate, this bond is after the phosphate, releasing phosphatidic acid and an alcohol. PLC and PLD are considered phosphodiesterase because they break a phosphate diester bond. Arachidonic acid is the precursor of eicosanoids. Eicosanoids are paracrine hormones, substances that act only on cells near the point of hormone synthesis instead of being transported in the blood to act on cells in other tissues or organs. Arachidonic acid undergoes enzymatic oxidation and it yields various different products. The first one, prostaglandins, contain a 5-membered ring and as opposed to arachidonate, as you can see. And prostaglandins are involved in inflammation and fever. Arachidonate can also get converted to thromboxanes. Thromboxanes have a 6-membered ring containing an ether functionality, as you can see here. They're produced by platelets, also called as thrombocytes, and act in the formation of blood clots and reduction of blood flow. The third type that arachidonate can get converted is leukotrienes. And leukotrienes, as the name suggests, are found in leukocytes containing 3 conjugated double bonds. Leukotrienes are involved in smooth muscle contraction in lungs. The arachidonic acid pathway is a target of many steroidal and non-steroidal anti-inflammatory drugs, or NSAIDs. Now, in the arachidonic acid pathway of inflammation, what happens is a membrane lipid, upon inflammation, a signal is sent to the cell, and that signal is perceived by cleaving the membrane lipid into arachidonic acid, and this is catalyzed by phospholipase A2. Steroidal hormones like corticosteroids block this pathway, and this is the earliest pathway that corticosteroids block, and hence they are really powerful. Arachidonic acid can then activate lipoxygenase or cyclooxygenases. These are enzymes that further trigger inflammatory pathways. And there are drugs like Zyflo or xylitol that targets lipoxygenase, or other non-steroidal inflammatory drugs that target cyclooxygenase. And there are other drugs that are downstream, act downstream, and stop inflammation from happening. But in a nutshell, what this slide tells you is that arachidonic acid pathway is important for inflammation. And there are drugs that can target these pathways.